We've created these mosquitoes that express Cas9, and that Cas9 functions very efficiently. You want the mosquito to do all the work, really. We targeted genes that would be important for sort of, vision or flight. We targeted a gene called ebony, which gave us a black body cuticle. We targeted a Hox gene known as deformed, which is important for developing facial structures. We targeted a gene called vestigial, which is important for wing development. And we had mosquitoes that couldn't fly. If you mutate this gene called yellow, you get a complete yellow body cuticle. It's such a striking phenotype. And one utility for that could be if we linked that gene to the male determining locus in the 80s, then males could have a dark cuticle, whereas females could be yellow. That could essentially be a way of genetically sexing them. And some of the more severe phenotypes, like the triple I mosquito and the mosquitoes that couldn't fly because we knocked out vestigial, we tried breeding them with each other and we couldn't maintain the stocks because they were just really unfit. So we tried to target genes which could be useful for controlling mosquitoes in the future. And we weren't sure what kind of phenotypes we would find because these genes haven't been targeted before in the mosquito. So it was all kind of exploration. Let's imagine if you wanted to go to Puerto Rico. Puerto Rico has a huge problem right now with Zika and dengue, and they don't really have a good vector control program, but it's a small island. One could go out and put traps and figure out, okay, well, there's 1 million mosquitoes. If you knew that number, then you could actually calculate, if I release X amount of mosquitoes, then by this number of generations, the entire population will be eliminated or replaced with resistant mosquitoes that can no longer transmit Zika or dengue. So you want something that's catalytic, where you can just release a small number of individuals and have a wide, long-lasting impact and actually solve the problem. There hasn't been a lot of characterization of genes in mosquitoes in general because the technologies to do that just haven't been there until CRISPR. CRISPR advanced the way in which we can target a genomic location and actually build these homing-based gene drive systems based off of CRISPR. They estimate that there's about 1,000 children dying every single day of malaria. And I have kids, and if my kid got bit by a mosquito and came down with malaria, how would I feel? I think there needs to be better solutions for controlling mosquitoes.